Good afternoon, ladies and gentlemen. I was once taught never to start a talk with an apology, but this time I have to. I apologize, I have to speak in English. So partly it's because this is not my most familiar topic, like lung cancer, EGFR. Trying to use Mandarin is a little bit tough. Now, I start off by asking a very simple question. Why do we do clinical trial? So I just searched the internet, and interestingly, I found Bill Gates got something to say about it. He actually just said the clinical used to determine whether new biomedical behavior intervention is safe, efficacious, and effective. And I focus primarily on the efficacious and also effectiveness at this point. So we have done clinical trial for many years, and those are the four cornerstones that we talk about. Phase 1 on the dose toxicity, Phase 2 efficacy on the selected tumor group. Phase 3 is a compare with the standard, and Phase 4 is on general population post-marketing study. That is our foundation, and I'm given the task to talk about this particular cornerstone. But then I looked into some of our success. This is a very famous study in lung cancer. Oh, by the way, I'm going to talk mostly lung cancer because that's all I know about. This is called FACS. This is one of the two studies in lung cancer that had a positive overall survival in the past decade. And this is the one that was published in 2008, initially in ESMO, and then in 2009 in Lancet. But today, where we with this cetuximab, just a few months ago, they give up. Merck alone saying they forget it. They are not going to deal with the EMEA anymore. There will be no further development of cetuximab in lung cancer. Why? A positive study like this one with some median progression-free survival. Why was there no development? Now, I think just come back to some of the basic concepts of Phase 3 study on the Cornerstone. First of all, we assume all patients are enrolled with the similar characteristics. By the way, these are the so-called routine standard thinking. Second, we actually kind of assume that two arms after stratification, they will be equal. Thirdly, we also assume whatever happened in the first line is going to impact the overall survival no matter what happened after. And also, we also assume there is minimal crossover. But are all these four factors true? My answer is maybe, unlikely, and maybe. Those are the few important assumptions we make when we do a Phase 3 standard trial. But then, some of these assumptions are not correct. So now faced with this kind of situation, how do we make it better? So I'm going to share with you my very simple mind of 5 steps to do Phase 3 design. First question, who may benefit? Second question, better than what? Third question, balance or not? Fourth question, better in what? Fifth question, how many and how much? Let me start off who may benefit. Now, going back to this FACS study, which is the combination of chemotherapy with cetuximab versus chemotherapy in unselected population, do we ask the question who may benefit? But if they go back retrospectively, subgroup the benefit, patient with skin rash benefit, but you can't do it prospectively. So we are faced with this situation. The simple answer to that is the fact that patients are heterogeneous. But then to ask the question, we have to ask first, who asked the question? Because sponsor, investigator, and regulator ask a different question. Now, sponsor, usually pharmaceutical, ask the question of who may benefit, but what in the bottom line is that the bigger, the more, the better. So the more patient benefit is better for them commercially. Investigator, we try to answer the question more scientifically is that who may benefit according to what we know. Regulator, they probably look into safety and also, in a more global picture. So when you ask the question, we have to think of these three things, but there are interactions about these three parties. Sponsor listen to the regulator, investigator sometimes listen to the sponsor, and then investigator sometimes complain about regulator. So there's all this interesting interaction going on before a decision is made on who asked this question. But most important, in my mind, patient asked this question. We have to ask the question from the patient point of view, which patient benefit? As a patient to see a drug, they will ask, am I the one who benefited? So I think from the investigator, from any of this party, I think that's still the fundamental asking question. Lung cancer, many failure. On unselected population, many trial, many targeted drug, all negative. But I think the answer is very simple. They're unselected population. We just learned recently, 2012, tumor heterogeneity exists within tumor, between tumor and lymph node, between tumor and metastasis. So when you're in face of heterogeneity, it's very difficult to assume the fact that between patients going to be homogeneous. So based on this situation, I borrowed a few slides from Lillian, who just gave an excellent talk in Hong Kong recently, is the fact that when you look into the different cancer type, you get similar kind of mutation, but then the distribution is different. So some cancer are more predominant in certain type of mutation, some not. But then again, tumor type, specific patient has to be different. And we do look into some of the drugs that have been approved, in the recent decade and a half. You can say that most of these drugs that's approved are highlighted the one that they are the one with the driver oncogene. So majority of this so-called molecular target therapy is associated with a well-known target oncogene and a very specific inhibition. Now, there are some situations like the HER2 with amplification, but it's likely a driver oncogene, but then it's not a mutated one. But then, certainly that, the efficacy is also improved with the trastuzumab or lapatinib. And also, important thing is that out of this one, how many of them does have biomarker? Well, again, Lillian has shared with us is that only about half of them had a biomarker. But I also asked further, who are the ones who don't? The ones who don't are actually the one with anti-angiogenesis, mTOR inhibitor, and immunomodulator. But most of the growth signal, actually, we can find a biomarker. So under these circumstances, if you go back to lung cancer as a model, the first thing to do a phase 3 clinical trial is the fact that we have to understand the oncogenic distribution. Adenocarcinoma, squamous cell carcinoma alike, we have to have a certain understanding before we can ask who benefit. We have to ask who is the patient with the particular oncogenic driver. So based on this assumption, how do we proceed? Well, there are a lot of difficulty in a sense is that the patient or population are so diverse. So I think almost every single population may have to have their own genomic profile. This is data from Wu Yilong, and that he actually already showed that in some of his other presentations. But you can see that even within Chinese population, different characteristics, the histology, the smoking status is going to change the distribution. So this has a very clear fact that whenever we approach a population and decide who benefit in a phase 3 study, we have to understand the different genomic profile between ethnicity, between different genomic profile between smoker and non-smoker, and also histological type. So those are some of the fundamental information you have to have on hand before you can design a phase 3 study. If you don't even have the basic information on the genomic profile, don't even gamble on it. You'll be just like going back to what we used to do. Now, so these are some of the design that David Gandera showed in his recent publication in clinical lung cancer. This is the old way. We got preclinical phase 1, phase 2, and phase 3, and then the time it takes may be long, 7.5 years as you calculate. But this time is the fact that if you know a biomarker, we should design our study in a different manner. No longer using the old model of phase 1, 2, and 3 as state. But certainly in the phase 3 design, in my mind, there's 2 that we can adopt on the selection process. In the biomarker selection is either you clinical pre-select them and then you do retrospective biomarker, or you can do the biomarker selection upfront. I couldn't think of the third way, in a sense. You can mix them, but then those are the 2 basic principles. You either know them or you don't know them. You don't know them, you have to enrich them so that you maximize the chance of finding the biomarker. So first of all, going through this, the so-called the biomarker one, that you actually look into the situation that you have a population, that you got the biomarker, and then you have the positive and negative population, and then you randomize them, and both of them receive it. And then on the other hand is the fact that you have to have only the patient with the positive and randomize them. But when you have those times, you have to face 2 questions. First question is that how specific is your biomarker? Now, if it's a mutation, it's easy. But if it's a semi-quantitative, like IHC, like FISH, you have to understand the threshold problem. The threshold is artificial. Wherever you do the cutoff to call it positive, negative, is quite artificial. And second thing is that you have to understand the preference of this particular biomarker before you proceed. Without this two bit of information, then you're again gambling. So these are some of the biomarker design as suggested by David Gandera, but I want to highlight this on 2 points. First is the fact that it's the strength of the knowledge of the biomarker cutoff. So the more you understand that, the more specific that you can be. But then also you have to look into the preference. Is that the lower preference, then you probably have a high understanding of this definition of cutoff, then you can use the biomarker selection model. But on the other hand, if you actually have no specific information about the cutoff, but the preference of this positivity is high, then you want a more generation population so that you can learn a little bit more in the process. So, and through this is a paradigm shift from one to the other. But in the back of your mind, you look for the strength of knowledge on your marker, and second, the preference. But then, now I'm just going to focus something that I know the best, which is the IPAS, which is actually somewhere here. Here, we kind of know that the biomarker is quite specific because of the mutation. But, and also, in the Asian population, we know that the preference is moderately high. So for that, in this IPAS, that I specifically want adeno and never smoker. Basically, we kind of estimated that the mutation rate has to be above 40 to 50% before we proceed. And I know that if this study, if the mutation rate is less than 40%, it's likely going to be a negative overall study. And secondarily, when we look into this data, once you see this curve, likely that you have two specific populations, one with the mutation positive, that TKI is superior to chemotherapy, and the population that mutation negative with the chemotherapy better than the TKI. And then you use your retrospective sample to validate your hypothesis. So you have to collect a substantial amount of sample. This day, you can probably ask for 80-90% of the sample. But in my old days, in 2005 or 2006, we were able to obtain about 34%, and that was already the highest tumor sample recruit rate compared to other lung cancer study. So there's no doubt sample collection retrospective is feasible. And then with that, we were able to demonstrate so-called this curve, showing that the mutation-positive population, that the chemotherapy is inferior to a TKI, and the mutation-negative population, the chemotherapy is better than the TKI. Now, but more importantly, is that through this process of selection that we have to validate your biomarker. Because it's a dynamic process. Most of the clinical trial nowadays in phase 3 is a combination of the validation of biomarker and doing the trial at the same time, making it very challenging. So, for your understanding, retrospective can only give you a little bit of signal, can never give you any so-called absolute validation. Prospective single arm can give you some idea, but mostly is on the prognostic. Only on the prospective randomized study, you got both the standard arm and the treatment arm and have the biomarker, then you may be able to validate the study by the interaction test. Now, let me just explain the value of doing a clinical selection such that you get an interaction test. So if I'm wrong, just please correct me, okay? Or say nothing. Don't tell me that I'm wrong. So, study population. Generally, you randomize to A and B. But within this population, you actually have a population with the biomarker, that receiving A and B, and also patients without the biomarker receiving A and B. With the patient, with each group, then you actually have a hazard ratio. If presumably your biomarker is predictive, your hazard ratio should be less than 1 in your patient with the positivity of biomarker. And then the hazard ratio should be more than 1 if the patient without the biomarker. So the interaction test is mostly on the interaction between these two hazard ratio to look for their so-called the p-value in the statistical significance. So we must have both group of patients within the same randomized study before we can validate a biomarker on its predictive value by interaction test. If you go back to the IPAS study, you can see a 0.48 in the positive group, and the hazard ratio for the mutation-negative group is 2.85. As a result, you have your interaction test of p-value less than 0.001. Am I doing okay, Jack? Yes. Okay, thank you. Oh. And so that is the interaction test we try to achieve with this kind of study, using clinical selection, enriched population of biomarker, such that we can calculate the interaction test and validate the biomarker. But this is not always possible. In a sense, that for some uncommon mutation, you don't want to do that, or less common mutation, you want to do that. So you have to use the positive group, so-called the biomarker selected study, and which is actually this spectrum of this diagram. Now, the most classic one, favorable one, is the most recent 1007. We learned from the phase 1-2 study that crizotinib had high response rate, and the drug was approved based on a phase 1-2 study. So therefore, you select the patient with crizotinib, patient with the M4L translocation, and then randomize them to crizotinib and pemetrexed, and use progression-free survival as a primary endpoint. Very clean-cut biomarker, and the result, you got the result highly positive, 7.7 months versus 3 months. Absolute positive study, won a lot of applause in ESMO this year. And the reason is that because we have very clear-cut qualitative biomarker. You either have mutation or you don't. And also, we can use it because there's no preference of a biomarker because it only occurs about 3 to 5% of all the lung cancer population. So this is the way to go when you have a situation like this. But we don't have M4L all the time. What if there's something else like CMAP? So I quote you another example. This is a randomized study comparing to ertilip and MAP versus ertilip. Then they start off with a so-called the general population, but then they use a biomarker called the IHC diagnostic positive. Considering the fact that they use IHC distinct for the CMAC expression, when they're over 50% of the cell, they have 2+ or 3+, they call it MAC diagnostic positive. And with the diagnostic positive patient, you can see there's an improvement in both progression-free survival and overall survival. So the company get very excited. And so now they're going to do the phase 3 study, 480 patients. Every patient got screened and look for this MAP diagnostic positive, and then randomized to this 2 arm as if the same in the phase 2 study. It's a good study, makes sense because they're phase by phase 2, but what's wrong in phase 3? I think go back to that. Is this a clear-cut qualitative biomarker? 50% is just because the guy say 50%. There's no biological reason to say 50%. It is a matter of convenience in a way. It's a matter of what they decide in the beginning. So when something is wrong with your biomarker cutoff, the whole thing may collapse because the 60% may benefit, maybe the 40% benefit. You do not know how wide the margin is. And second is the fact that the good thing about this study is that you will end up with about 40-50% of patients to be positive. So therefore, you got a pretty good preference rate and help you with the study. So, but again, I put this as an example is that every time when you design a phase 3 with biomarker selection, think about the biomarker, think about the preference rate before you decide how to do with it. And then finally, is the limitation on this design is the positivity is limited to your so-called the knowledge about it, and also the testing method has to be very efficient. Now, if this testing method is very complicated, unique to certain center, when you go in a multi-center randomized study, it's almost impossible. And again, one problem with this design is that we will never find out what happened to the biomarker negative population. Especially with this kind of circumstances, is that once you do not know about negative situation, you actually may be potentially giving up a good drug for quote-unquote the biomarker negative group. Second part, better than what? That is the control arm. Now, there's 3 different so-called control group. One is that you can use a study group. Now, that is such like the ECOG. ECOG say Taxol, Cabo, bevacizumab is the standard for everything in lung cancer. And this causes a hell of a lot of pain. But why do they do that? Well, because of the 4599. So they believe their doctrine. They believe everything they say, which is, we know in life, that's not quite true all the time. So there is a problem when you hold on to a certain doctrines is that because my group have done that and show that, therefore it will forever and ever, amen, that is the study group. Sometimes there is a problem. Approval agency is important because we have to respect what they say. But again, and sometimes it can be changing from EMEA to FDA, SFDA, their control could be different because every country have a different standard of their therapy. So they may recognize different therapy as their control. Thirdly, you can look for the guideline, and sometimes NCCN is pretty safe guideline for you to use as standard because it is so-called a peer review situation. But again, every single time when you decide on your control group, think about these three groups of people before you decide. Now, let me just go back, once you try to do the design group, what is the problem? Now, these are the famous lung cancer study comparing the TKI in a mutation-positive patient with a control arm. But look at the control arm. IPAS, we use taxol-carbo, and then a Korea gemcis, dorsal-cis, taxol-carbo, gemcis, 1 or 4 regimen in Spain, and PAM-cis in the Luxembourg 3. Now, of course, there's a Jones Schiller study saying that they're all pretty well the same, but we know that it's not exactly the same. PAM-cis is probably maybe better than gemcis in adenocarcinoma, and all this patient adenocarcinoma. And also, all this decision process always have a story behind it. Like, for example, the taxol-carbo. AstraZeneca was still thinking about United States when we designed this trial. They want to use taxol-carbo as the control arm. But look at the outcome. The response rate can be variable from anywhere from 18% to 47%. In a study, phase 3, when you have the change of the 10% in your control rate, it makes a big impact on your study arm in terms of success. I hope they're not complaining about my talk. And then there is also the median progression-free survival. It's also ranging from 4.6 months to 6.7 months. So you can see that the choice of your control arm make an impact. So think about it before you proceed. And then some of the factors that may influence you, you can kind of shift it, manipulate a little bit to favor your study. This is the sample size. The bigger the difference, the smaller the sample size. So you use a weaker control, then you can get a bigger difference. And also, you also want to look into toxicity and quality of life. You want your arm to win, you get a more toxic treatment, then you may win in the toxicity on this side. And of course, the cost means a lot as well. Now, the most classic example, is the fact that if you use bevacizumab as your control arm, it costs tremendously more to your study. Is it necessary? So you have to argue, fight, and say, forget it. You don't want your bevacizumab in your control arm. So all these that come into play that influence your final decision on what the control arm is, but those are the practical things you have to think about when you do that. Third, balance or not, that is your stratification factor. Let's look at Lux-Lung 1. Lux-Lung 1 is a trial of a 2-to-1 randomization of afatinib to the control in a 2-to-1 fashion. Now, the stratification is actually using ECOG and gender. I start to scratch in my head, why the hell does gender got to do with the outcome? Why do you need to balance the gender when you look comparing a TKI with the placebo control? The ECOG makes a little bit sense, but then whether there's a major difference between 0, 1, and 2 in these circumstances is also a little bit debatable. But so, as a result, you can see that, yes, they balance it really well. But what are the key factors that influence the outcome of this study? Actually, how they respond to the prior TKI. Now, it's lucky the fact that they are also balanced, in the prior exposure to the TKI. But there's no guarantee the two arms to be balanced. Imagine their prior experience with TKI is different. It's going to influence the outcome of this particular study, because this is, after all, a TKI-related study. But most important thing they did not balance is the number of patients with TKI mutation. Now, why do I say that? Look at this one. Now, when you look into, this is the waterfall curve on the response rate, you can see that the mutation compared to placebo, it doesn't really matter on the mutation mutation, because, in a sense, if there's no treatment in one arm, you will not expect the outcome. But then the mutation affects the overall survival, which is the primary endpoint. You can imagine this as a study on the TKI. The patient actually may have further exposure to TKI depending on their mutation status. If you do not balance between the two arms, how can you be sure the overall survival is a reflection of the drug, but not a reflection of the mutation status? So those are the problem, is that in a stratification, they did not make sure the two arms are balanced, and they did not have any biomarker translation. So therefore, use of single-agent afatinib in approval process of the third-line situation is very difficult at this stage with this study. So some of the basic rules when you think about stratification. Number one, not too many. Only think about the relevant ones. Number two, has to base on biology. When you look in the study of TKI, you got to balance the TKI EGFR mutation. You have to have relevant biology behind that. And also, you have to think about how does this stratification impact on subsequent line of treatment, not just on the study treatment. So all those are very important factors to think about when you come to a stratification. Okay. Now, but then it's two different dilemma when you try to do stratification factor, in my opinion. When you do clinical selection, meaning that you select the patient clinically, you do not have the biomarker. Your stratification objective is make sure the biomarker are balanced. But once you select your patient by biomarker, you don't need to do that anymore. Then you select the stratification factor to make sure the prognostic factor are balanced so that they can be equal in both sides. So in my mind, when I choose a stratification factor, those are the two important factors that I consider. Now, let's just go through some of the example. This is one study that we have presented in ESMO using a Fagutuximab, which is a HGF inhibitor. And it's a combination of this drug with gefitinib versus gefitinib. Now, look at it. I use an IPAS population, Asian adenocarcinoma, leiomyoma smoker. So, in other words, I use clinical selection. But then, I have to make sure that both arms have the balanced number of EGFR mutation. So, therefore, I add on the smoking history and the gender, because this time, gender make a difference in terms of incidence of EGFR mutation. So, as a result, in my study, they are pretty well close to balance in the mutation, 35% and 40%, which is acceptable, equal of the two arms. But then on the LUXNUM3, which is already patients who are known to be EGFR mutation positive, and then they choose 19 stratification by 19 or 21 because there are some difference between 19 and 21 in terms of prognostication. As well, they choose Asian versus non-Asian. One reason is the prognostication. The second reason is that they actually plan a subgroup analysis according to it. So therefore, they want to make sure both arms are balanced. So there has to be a reason when you choose your stratification factor in a phase 3 study. Last but not the least is, not the last, second last, sorry, I'm running over time a little bit. Better in what? This is taught about PFS over OS. I just give you a short version of what I'm thinking, especially using lung cancer as an example. So when you originally look into EP, is standard etoposide cisplatinum as a prolongation of survival from 6 months to 8 months. We're talking about chemotherapy, reducing response, and then the tumor gets stable, and then eventually progress, and then the patient eventually die. So the component of that is that there's a progression-free survival and overall survival. This is the basic where we start off. And then we have the second generation chemotherapy like GemCis. Now, so what happened is that we actually have the longer progression-free survival, and therefore we end up with longer overall survival. That's how we learn, the improvement with the second generation. But then after that, we got things more complicated. We got second-line chemotherapy. Second-line chemotherapy, docetaxel, we actually have a second progression-free survival to end up a summation into longer overall survival. So the important thing to consider is that the overall survival could be just a representation of 2 or even more of prolongation of progression-free survival. But to make life more difficult is the fact that patients are heterogeneous, some with mutation positive, some with mutation negative. So mutation positive patient, when you put on a TKI, you get a very prolongation of progression-free survival. But then on the patient without mutation, you only get a little prolongation of progression-free survival. So when you look at BR21, there is a positive study overall of the overall survival. Ertilib has a ratio of 0.7, and that drug got approved by FDA and EMEA. But as a matter of fact, this is a heterogeneous population. We have two populations, one with a long progression-free survival, number 2, a second line, and one with a tiny progression-free survival, number 2. And then you're mixing them together before you end up with your overall survival curve. So keep this concept in mind whenever you decide that when you have a heterogeneous population of this looking into overall survival, you've got more than 2 group of patients with 2 different types of progression-free survival. So the third concept that you have to hold in mind is the fact that the crossover. In all this lung cancer study, you can see that none of the overall survival is positive. The reason is the crossover. The IPAS, no difference, and you can see the crossover rate is 65%. And then in other Japanese study, as you can see, there is a change of the so-called the survival over time, but you can see that the crossover rate is about 98% in the chemotherapy arm, and then for the Gefitinib, the crossover is 65%. So therefore, there's no way you can demonstrate overall survival benefit. So when you choose a final endpoint, you have to put all these factors into consideration. There's so many variability and possibility, but you have to keep this idea in mind before you decide which is the most appropriate endpoint. Last but not the least is how many and how much. How many is a question of sample size. I leave Jack Lee to talk about it. I don't want to talk about it. But once Jack tells you how many he needs to the sample size, you go back to look at your wallet. First is your patient resources. Can you attain that goal? For that, you must again go back to understand epidemiology of the biomarker selected population. If it's a rare or uncommon or not population. And also you have to look into diversity such that you can decide which is the population you want to choose. So that you can decide on the number center and see whether that goal is realistic or not. How much? I always ask the question, is it a registrational study or not? Is it a registrational study, pharmaceuticals going to make the money eventually, they have to invest. But once I come to investigate initial study, I cut the cost as much as I can because they won't keep any money and I don't have any money. So basically, I put all those factors into place on how much and how many before I find the sample size. And then the last part of my talk is actually the supplement to what Yilong had talked about, is the so-called the cluster design. Nowadays, a single biomarker, we are lucky enough to pick the low-hanging fruit of EGFR mutation. Everything else will be more difficult because they are more uncommon. Now, EGFR, a big segment of the pie, you take it out, sufficient to do your biomarker selection study. But you look into everything else add together, that is almost same as the EGFR mutation. So if you do it one at a time, it's very difficult. The question is that can we do them together? Now, to get them together, you can do the genomic profiling, just like what Yilong has suggested. You profile the patient according to your ROS, your RET, your PI3K, HER2, BRAF, and CMAC, and then you match them with the respective drug. So in a single arm study, that you just profile the patient, know the genomic marker, give them the right drug. That sounds very good. And then we decided according to the response rate. With every single of this one, you are going to end up with a response rate. To me, if it's an oncogenic driver that worth your investment, I will say that you must have a response rate about 50-60%, if it's an oncogenic driver. If you got 15%, remove it. 20%, remove them. Just retain the highest one. And then for the highest one, can we do the randomized control study? You profile a patient to look for this 3 gene and then give them the drug accordingly in a 2-to-1 randomization. And upon the progression of the standard chemotherapy, you provide the drug back and using progression-free survival primary endpoint. Now, how do we do the sample size? I don't know. Is it practical? I don't know. So for this to be practical, I think that we have looked at what are the pharmaceutical involvement. If you want to do this design, those are the 6 pharmaceutical involved. Can I ask 2 pharmaceuticals work together? Tough. Ask 6 is almost impossible. So I think this is ideal, but somehow I think the people who can tell them to work together are the regulator. If the regulators say, this is the way to go, I'm sure they probably will start listening. So I think we have to overcome a lot of barriers if we want to go this direction. It's number one, we've got to get them to talk to each other. We have to overcome a lot of legal barrier. And then we probably need to have a study collaborative group like CETO to be the leader and then to head the whole thing. And we probably get acceptance by SFDA, FDA before we can move this kind of concept onward. So, it's happening. I don't know about this. And again, I learned this from Lillian, that this is happening in Focus 4. Something very similar to colorectal cancer, BRAF, PI3K, KRAS, and then on the wild type, and then they do a randomization accordingly. And in Lillian's place is doing something along the line of the genome profile every patient, and then they assign the drug to the patient according to the genomic profile in a phase 1-2 setting. So I think the paradigm is shifting to this kind of direction where we have to work together such that we can provide the drug to the patient accordingly, according to their genomic profile. So to summarize is that, these are my very 5 simple steps. Full benefit, better than what? Stratification on the balance or not, better than what? Progression-free over survival, and how much and how many. And I hope eventually our new design is better than our old design. Thank you very much for your attention. Thank you.
So just now, Dr. Tony Moore gave us a wonderful speech. Any questions and comments, please? Okay, Dr. Wu.
Tony, excellent talk. So as you know, for the advanced disease, so this belongs to the no cure disease. So in all the guidelines, for the advanced cancer patient, the goal is to prolong the survival and the quality, not the cure disease. So I think that for the phase 3 trials, we can using new the endpoints such as the symptom improving and the symptom deterioration as the endpoint. Because these patients will also live longer and also quality of life. Yeah.
Well, I think those can only be supplementary because number one, although the validation process for all those questionnaires are very strict and high quality, but it's still a highly subjective situation. I feel better, because, my mother gave me a glass of milk today. There are so many different factors that influence your quality of life apart from the drug itself. So I don't think we can use just quality of life alone as a primary endpoint. So at this moment, I think if it's an oncogenic-driven situation, it's the fact that you have to use the quality of life information to supplement and your prolongation of progression-free survival to make this a complete biologic story. But then I personally would be very reluctant to just use a PRO as the only and primary endpoint for registration of the drug.
And just now, Dr. Jack Peng submitted a question, how to promote the collaboration between ESCO and CISCO and ISMO or some other professional society. I know, act as the Vice President of CISCO, you in charge of international affairs with Dr. Shun Lu. So what's your opinion? You may talk about that.
Well, right now, ESCO and CISCO are mostly educational society. I know CISCO is starting to look into some of the clinical trial, but ESCO, to my understanding, has so far no specific plan to go into clinical trial. So I think we're talking about very different body of service. We can do this kind of stuff, which is educational. But for us to function as a group in clinical research in the future, I think we still have a very long distance in that in such regard. But I can see that there are a lot of possibility in training. For example, we already some initial effort between ESCO, CISCO, and ESMO in terms of so-called sending off to oversee for training, which may bring back idea and collaboration ability. But at the end of the day, is that trying to do a intercontinental kind of collaboration on clinical trial, I think is still very difficult and challenging.
Just to add to what Tony said, I think that's essentially very much true, focused on education. But I think there might be opportunities in the policy realm where Rich was talking about facilitating that kind of dialogue with regulators and other policy issues, and where there's sort of commonality of concerns or sharing of approaches or barriers, and highlighting those barriers and facilitating that dialogue might be another area of collaboration between the professional societies.
for the phase 3 confirmatory clinical trial, for the last line of the patients, we will find a difference between the agencies to say that maybe for EU, the best available therapy as a control, but for US as a placebo used as a control. I like to know what's the thinking behind that, and also from the practical point of view, what's the strategy to use the best available therapy? Because there are many kinds of best available therapy. Maybe it's a single agent, a different single agent. Maybe it's a combination. Do we need to define several, or maybe one or two or three of the best available therapy, especially single agent, to use as a control? Maybe is there any other use as a suggestion?
Yeah. I think the difficulty that we're in a transitional phase of a biomarker selected design to from the unselected population design to a biomarker selection design. A lot of our control of so-called response rate in a population is an unselected population. For example, we use pemetrexed as a second-line chemotherapy for adenocarcinoma. The response rate in most studies is 9%. But when you look into the crizotinib study, the response rate is about 22%. So L-positive patient, the pemetrexed work actually well, and then you have high response rate than you expected. So the control, of selection is not biomarker it's not biomarker specific. So therefore, whoever agency, whoever guideline don't have the information on hand on what is the best therapy for this biomarker selected population. I think that's where some of the problem is, for our future onward. But now, as we go along, have more information. For example, EGFR mutation positive patient, the control will be gefitinib or erlotinib, depending on geography. So now the future study on the EGFR mutation positive patient can no longer be chemotherapy. Lung-3, well, actually Lung-6 will be the last study on mutation positive patient using chemotherapy as a control arm. The future has to be a TKI, and there are two studies coming up. One's a Lung-7, the other one is Archer-1050. Both use gefitinib as a control arm. So it's a matter of evolution, of our knowledge on the biomarker. Okay.
Is there any comments?
Dr. Lu Shun.
One question is that previous, the cytotoxic chemotherapy, PD is, we give up. But for the targeted drug, some patients, even PD, maybe still get benefit from such as the Resal-Tarceva or even ALK. So it's quite complicated for such kind of the situation. And the heterogenesis, some patients maybe still get a benefit, but some patients not. So for such kind of situation, how to design trials, it's much complicated.
Well, I think the registration agent will hate me because I want to say is that we want to get rid of RECIST criteria for molecular targeting drug, because I don't think it's the best one for molecular targeting drug, in a way of a single lesion, because there are so many things we can do if the patient have a new single lesion that come up. The RECIST criteria was actually originally designed for chemotherapy, and now the biology behind the TKI is totally different. So we have to find new way, and you probably know that we are doing some study on treatment beyond progression. We are also doing study on trying to redefine what is the potentially better definition of progression for TKI. So we are still on a very much a learning process, on the progression, the definition of progression on the TKI. Now, but that is different from the primary progression, obviously. What you're talking about are the patients who previously respond and eventually progress. And I think we had to find better definition. And as a result, our new progression-free survival have to be redefined, which will make things very complicated.
Okay, yes.
Just to reassure you that we don't hate you at all for saying this. I think RECIST have a lot of merits, but they don't go beyond what they were initially designed to achieve, which was some type of sign of activity in phase 2 studies for solid tumors. The fact that we use progression-free survival based on RECIST as a primary endpoint of phase 3 trial, that's the challenging part. But RECIST was never designed to do that. And we are all for trying to come up with better definitions of progression. If you use it like the FDA seems to be using it as a reasonably likely surrogate, then you can accept some imperfection in the surrogate. But if you use it the way EU is using it as a sign of clinical benefit, then it seems almost a paradox that in this definition, you exclude clinical progression. It's absurd. You're saying this is of benefit to the patient, measures direct benefit, but it should only be based on radiological criteria.
Yeah. Do you think it's possible to organize something in the future with different regulatory body and, probably the RECIST criteria guys, and so that we can actually try to find a new definition of progression for TKI that's acceptable by all parties? Do you think that this kind of action will...
Yes, I think it's long overdue. We are working on it, but we are a little bit isolated, and we should have that discussion and also talk with other regulatory authorities and see how much they are willing to venture into a better definition of progression.
So do you think organizations like ESCO and Cisco will be a good medium to kind of glue this together?
Absolutely, any type of platform within the, let's say, the scientific world, because these are scientific discussions and not regulatory discussions.
I'll put that on my agenda for March 1st. Okay.
Okay.
Yeah.
Okay, we probably should move on. Okay, let's take one more question.
Last question.
Professor Mock talked about the third phase research, but we know that for the third phase trial is based on the positive result of the second phase. So if concerning placebo, will the use of placebo affect the benefit distribution of this patient, because those patients who receive placebo will not receive any benefit at all? So how do you view this phenomenon? Well, I think it is based on what kind of biomarker you decide to pursue. So if you use placebo, you have to make sure of the crossover. But it is not for the biomarker driver. Just for the pure comparison, it's another case. So I think we need to consider the situation on the base cases, on the case-by-case basis.